All right, good day, grade 12s. Welcome to today's bio lesson. Today we're going to deal with dihybrid crosses and probabilities. Now we are going to be turning our attention to not just looking at one trait, but actually looking at two traits at a time and predict probabilities of offspring having two traits that we happen to be looking at. So a one trait cross is called a monohybrid cross. So when we're looking at two traits at the same time, we're looking at a dihybrid cross. All right? So to understand the crosses, we must remember a couple of things. The law of independent assortment. So remember when we talked about mitosis and meiosis, the chromosomes are lining up on the equatorial plate in metaphase, and they are lining up independent of each other. All right, so when we're going to work with these following traits, we're assuming that they are on different chromosomes, and that just because you happen to have one trait for one gene does not mean that you would get another trait from a different gene. We're assuming that they are on separate chromosomes and that they will independently assort. So that's one of our assumptions. Okay, now guys, when we set up our dihybrid cross, some things are going to look a little bit different. So in this question, we're going to be looking at the seed, whether it's round or wrinkled, and its color, whether it's yellow or green. And we're going to be looking at both of those traits at the same time. You'll notice that when we do the genotype for a dihybrid cross, we need 4 alleles, because remember, you always get 2 alleles per trait. So we're looking at 2 traits, so we need 4 alleles. And we're going to keep the alleles together for the same trait. So here we have a pure breeding, round yellow plant. Pure breeding, homozygous dominant. We're crossing it with a pure breeding, recessively trait plant. So it has both recessive traits. It's wrinkled and it's green. So little r, little y, little y. So those are pure breeding plants. So we're going to put them on our parent line. So there they are, pure breeding plants. And the F1 generation, I hope you can see that the F1 generation, the only possible genotype we can get is big R, big Y, little y. Because this parent's only going to be able to give a big R and a big Y in its gametes. And this parent can only give a little r and a little y for its gametes. So this is the only possibility. But we're going to do a self-cross. So we're going to take individuals from the F1 generation and cross them together. All right? So when this happens, we have to set up our Punnett square. Now this is going to fill in for you, but just to do one of them, guys, we essentially need to predict all the possible alleles in the gametes for let's say this individual. So of course we could get, for this individual, we could get a big R and a big Y. And remember, you can only have one of each letter. So we can have this big R with this big Y, or we can have the big R and a little y. We could have a little r with a big Y. And lastly, we could get the little r with the little y. So there's 4 possibilities for different gametes for this plant. Now I hope you can understand that across the top for the other plant, it's going to be exactly the same. So we would go across and say, oh, well, this parent also provides the possibility of big R, big Y, big R, little y, little r, big Y, little r, little y. Whoops, I see what I did there. There we go. All right. Common mistake, besides what I did right there, a more common mistake is to keep the same letters together and have maybe 2 R's and then 2 Y's. And you guys, we can't do that because of the law of segregation. We separate those alleles. All right? Okay. Now this is going to fill in for us. So I'm going to, get rid of this. And it's going to be much easier for you to see in white. So there we go. So this Punnett square has now been filled out for us with all the possible genotypes for the offspring. And this is as big and as complicated as a dihybrid cross is going to get. This is a 4x4 Punnett square, and there's no way to make it any easier, really. All right, so now remember that the genotypes aren't really as interesting as the phenotypic ratio. So the next slide I show you is actually going to show you in picture form the results that we have. All right, so here we go. So here's in picture form, the results of that last Punnett square. So we can see, remember we had big R, big Y, big R, big Y. So that means some of the offspring are just like one of the parents, the pure breeding homozygous dominant parent. All right, and there's actually only going to be 1/16, one out of the 16 squares that would have this particular genotype. That provides round seeds, and they're yellow, round yellow seeds. But that's not the only way to be round and yellow, right? You can be round and yellow if you have 2 big R's, 1 big Y, and a little y. You'd also be round and yellow. So when you go through and you actually identify the phenotype for each seed, you're going to find that there are 9 of the 16 that are round and yellow. You're going to find 3 that are round and green. Here they are right there. 1, 2, 3. You're going to find 3 that are wrinkled and yellow. Where are they? Right here. 1, 2, 3. And you're only going to find 1 that has both recessive traits, wrinkled and green. 1 in 16. Now guys, it's not a bad idea to just kind of memorize that when you have a 4x4 Punnett square for a dihybrid cross, where all 4 gametes along each side are different, you always get what we call this 9:3:3:1 ratio. So it's not a bad idea to commit it to memory. If you commit it to memory, you don't have to ever really do one of these great big Punnett squares. But just remember that 9 means both dominant traits. 3, 1 dominant trait, 1 recessive trait, and then the next 3, we just switch the traits around so they have the dominant color and the recessive seed coat, and then 1, only 1 in 16, shows both recessive traits. Not a bad thing to be able to memorize that. All right. So, what if we cross an organism that was heterozygous for both traits, like we had before in our F1 parent, but this time we're crossing it with a plant that is homozygous recessive for both traits. Little r, little r, little y, little y. Well, guys, I want to show you, like if we started this off, right? We do have the 4 different gametes for our double heterozygous parent, because there's 4 possible combinations. However, there is only 1 possible combination for this parent, which is the little r, little y. So why would we go and like fill this whole thing out and have 4 lines that are exactly the same? I would simply stop right there and say that we have 4 different genotypes and then do the phenotypic ratio from there. So I don't think this one is done for us, so let's do this one. So the phenotypic ratio, this is a round yellow, and we don't have any more round yellows. So I would go 1 out of 4 round yellow. Then this is a round green, so 1/4 round green. Sorry if you can't see this very well on the screen. And then, just to continue quickly, we would have 1 wrinkled yellow, 1/4, and we'd also have 1/4 wrinkled green. So there we go. That would be the phenotypic ratio. We'd have that's predicted in the offspring of those parents. But notice how we don't have to do a huge Punnett square. We only have to do a 4x1. All right. Now, the next thing we want to talk about, guys, is probability. For instance, probability comes in really handy if we have, we might be looking at 2 traits or even 3 traits. A trihybrid cross, and we never, trihybrid crosses would get very complicated and very large. So what can save us some time is we do something called probability, and we use probability rules to make our lives easier. Now, probability is the study of the outcome of events or occurrences. And it's expressed by this formula. And you may have done this in math class. You may have worked with what's the probability of getting a jack of hearts, or in a deck of cards, what's the probability of flipping a quarter and getting heads twice in a row. So that's all probability stuff. And essentially it's a ratio. It's a fraction. It's the number of chances for an event over the number of possible combinations. So for example, if you were flipping a coin and you wanted to know what's the probability of getting heads with one flip, well, you're flipping once, so you only have one chance to get heads. And what are the number of possible outcomes? 2, heads or tails. So your probability would be 1/2, 1/2. So of course, our phenotypic ratios are essentially stated as probabilities. So like I said, in genetics, we use probabilities to predict the likelihood of a specific trait or traits showing up in an offspring. So to determine the probability of an event, there's 2 important rules. Number 1, the rule of independent events, which simply says that one event doesn't have any bearing on another event. So for example, going back to flipping a coin, if I said, what's the probability of flipping heads with one flip, that's 1 in 2. And then if I do that and I get heads, and then I say, well, what's the probability of me flipping heads again? Well, it's still 1 in 2, because the second flip has no bearing on what happened in the first flip. Okay, so that's the law of independent events, so that rule needs to hold. And secondly, there's the product rule. Now, the product rule, quickly stated, says that if you have 2 independent events and you want to know the probability of them occurring at the same time, you multiply. That's why it's called the product rule. Product is multiplication. So you multiply the probability of those 2 events together. So going back to coin flipping, if I said, what's the probability of flipping heads maybe twice in a row? Well, you'd multiply the probability of each one of those together. So we know that it's 1 in 2 for the first flip of getting heads. And because they're independent events, it's also 1 in 2 for flipping heads with the other coin, or the second time. If you wanted to know what's the probability of flipping heads twice in a row, multiply those 2 events together. 1/2 times 1/2, that'd be 1 in 4. You have a 1 in 4 chance of flipping heads 2 times in a row. Hope that makes sense. Now, in order to speed this up, let's look at an example out of the textbook to just speak clearly about what's written here. There's 2 traits we're looking at. We're looking at earlobes, and free earlobes, like mine, is dominant, so that would be capital E, and attached earlobes is recessive, little e. Then there's your hairline, and a widow's peak, where it's pointed here, is dominant, so we're going to use big W for widow's peak, and a straight hairline is recessive, so little w. So let's say we were looking at those 2 traits, and then we had a man who is heterozygous for both types, mates with a woman also heterozygous. What is the probability that they would have a child that has a straight hairline and attached earlobes? Now, I know we could do the big old 4x4 Punnett square and start to like dissect that and have a look at that. But what can often be easier is, why don't we look at each trait separately and then use the product rule to predict the combination of those 2 traits at the same time. So that would kind of look like this. Okay? So we're going to deal with each trait separately, and what we would do is a monohybrid cross for each trait. So we would look at the earlobes first, and then figure out what the probability is of getting the earlobes that are mentioned in the question, and then we would do the hairline, right? And then we would have 2 different probabilities. If we wanted to predict having them both at the same time, we'd multiply them together. So we're not going to see the worked out Punnett squares for those individual traits. Again, I'm kind of assuming now that you could do those Punnett squares if you needed to. So let's take a look at this situation. So what's the probability? Now remember, the couple is heterozygous for both traits. What's the probability that we're going to have a child with widow's peak and free earlobes? Well, if we did the Punnett square, we would find that for this couple, there's a 3/4 chance of having a child with widow's peak, and there's also a 3/4 chance of having the free earlobes. Remember, those are both the dominant traits, so typical Mendelian cross expects a 3 out of 4 probability for the dominant trait if both parents are heterozygous, carry the recessive allele. And so if we wanted to predict a child having both of those together, we would multiply those probabilities and we would get a 9 in 16 probability. Theoretically, if they had 16 children, we'd expect 9 of them to have both of these dominant traits. What about a straight hairline and free earlobes? Well, this time the straight hairline is only a 1 in 4 chance. It's the recessive trait. And the free earlobes, remember, is still a 3/4 probability. So again, multiplying those together, 3 out of 16. Same thing here. The widow's peak dominant hairline, recessive earlobes, 3/4 times 1/4, 3/16. And guess what? It would be 1 in 16 for the child to have both recessive traits. Okay? All right. Now again, we've got a question that talks about the first child from a mating of a male with free earlobes and widow's peak, heterozygous, a woman with free earlobes and widow's peak, having a male child with a widow's peak and attached earlobes. So again, if we did the little Punnett squares, we would find that just for attached earlobes, 1 in 4, because remember that's the recessive trait again. Widow's peak, the dominant trait, remember, would be 3 out of 4. This time we're also told we're expecting a male child. Well, there's a 1 in 2 chance of a male child. So the product rule says, hey, these are 3 independent events. We can multiply them together to get the overall probability. So 1/4 times 3/4 times 1/2, 3 out of 32 as your probability. Most often you would turn that into a decimal, especially if you were doing it as a test question, because it would be probably a numerical response. Okay? So that's the product rule. It can make I know it sounds complicated at first, but it can actually make your life relatively easy, and you don't have to get mixed up with really large Punnett squares. All right, so that ends today's lesson, guys. So make sure you might want to check out the Amoeba Sisters video on doing dihybrid crosses. All right, and yeah, happy studying.